Bismillahir Rahmanir Rahim. Histamine is among the first mediators of allergy to be identified. Its involvement in the modulation of allergic reactions was recognized as early as 1932, and since then studies have shown that histamine also plays a role in autoimmune conditions, gastric acid secretion, and hematopoiesis. Our video today will be about the biochemistry of histamine, and welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University. Histamine plays a key role in regulating various physiological functions, particularly in the body's inflammatory response. It's also integral to the pathophysiology of several inflammatory diseases, which give rise to the discovery of new histamine receptors and a deeper understanding of its role within the immune system. Histamine is found throughout all body tissues, with its highest concentrations in areas like the lungs, basophils, and mast cells. It acts as a potent vasoactive agent, affecting bronchial smooth muscles and stimulating nociceptive itch nerves. Histamine is a biogenic amine produced from L-histidine through the action of L-histidine decarboxylase, which relies on pyridoxal 5-phosphate as a cofactor. This enzyme is broadly expressed in different cells throughout the body, including those in the gastric mucosa, neurons, parietal cells, mast cells, and basophils. Basophils and mast cells are unique in their ability to produce and store significant quantities of histamine within granules, where it associates with chondroitin 4 sulfate in basophils and with anionic proteoglycans like heparin in mast cells. Recent studies have also revealed that various myeloid and lymphoid cells, including hematopoietic progenitors, macrophages, neutrophils, platelets, and dendritic cells, show histamine decarboxylase activity and can synthesize substantial amounts of histamine, although they do not store it like basophils and mast cells. Histamine is primarily metabolized through two major pathways: histamine N-methyltransferase, which accounts for 50 to 80% of its breakdown, and diamine oxidase, responsible for 15 to 30%, and only a small fraction, about 2 to 3%, is excreted unchanged. Histamine N-methyltransferase plays a key role in breaking down histamine within the airways, as it is expressed in the airway epithelial cells. Studies indicate that inhibiting histamine N-methyltransferase can lead to increased bronchoconstriction both in vitro and in vivo. Histamine N-methyltransferase is also found in the central nervous system, intestinal smooth muscle, small intestine mucosa, liver, and kidneys. Diamine oxidase is present in the mucosa of the small intestine, as well as in the liver, kidneys, eosinophils, placenta, and skin. On the cellular level, the effects of histamine are regulated through four distinct receptors: H1, H2, H3, and H4. These histamine receptors belong to the G protein-coupled receptor family, characterized by their 7 transmembrane structure that facilitates the action of various molecules. H1 receptors are GQ-coupled receptors that exert their effects through a signaling cascade involving increased phospholipase C activity, elevated cytoplasmic calcium level, and enhanced protein kinase C activity. H2 receptors are coupled to GS proteins, and their downstream effects result from an increase in intracellular cyclic AMP levels and the activation of protein kinase A. In contrast, both H3 and H4 receptors are G protein-coupled receptors, with their downstream effects mediated by a reduction in the intracellular cyclic AMP levels caused by histamine. H1 receptors are distributed widely throughout the body, found in neurons, smooth muscle cells of the airways, and blood vessels. When H1 receptors are activated, they trigger typical allergic and anaphylactic responses, which include increased itching, pain, vasodilation, heightened vascular permeability, hypotension, flushing, tachycardia, and bronchoconstriction. Also, H1 receptors play a role in regulating sleep-wake cycles, appetite, temperature control, emotions and aggressive behavior, locomotion, memory, and learning. They are also responsible for most histamine-related effects associated with asthma, which may include symptoms such as smooth muscle spasm, mucosal edema, inflammation, and mucus secretion. H2 receptors are primarily located in the parietal cells of the gastric mucosa as well as in smooth muscle cells and the heart. When activated, H2 receptors mediate gastric acid secretion, increase vascular permeability, and can lead to hypotension, flushing, headache, tachycardia, and bronchoconstriction. The clinical significance of H2 receptors remains ambiguous due to their limited measurable impact on airway function. When activated, H2 receptors can enhance the activity of the adenyl cyclase system, resulting in increased levels of intracellular cyclic MP. H2 antagonists have been recognized as effective treatments for duodenal ulcers and help prevent their recurrence. H3 receptors are primarily located in histaminergic neurons, where they regulate the release of histamine, dopamine, serotonin, noradrenaline, and acetylcholine in the central nervous system. The discovery of H3 receptors has been critical for understanding the complexity of histamine-mediated systems. Activation of H3 receptors can also inhibit neurogenic sympathetic vasoconstriction in the nasal mucosa. Also, H3 receptors serve as vital modulators of histamine release from mast cells and cerebral neurons. H4 receptors are found in the bone marrow and the peripheral hematopoietic cells, where they contribute to the differentiation of myeloblasts and promyeloblasts as well as chemotaxis. As a G-protein coupled receptor, H4 receptors are closely related to H3 receptors, with their messenger RNA being most abundant in the spinal cord. H4 receptors are often referred to as the immune system histamine receptors due to their significant involvement in inflammatory disorders and autoimmune diseases. Histamine receptors are also important for immunomodulation. They affect the activity and differentiation of T cells, B cells, monocytes, and dendritic cells in lymphatic organs and tissues, particularly during allergic inflammation. For example, T helper 1 cells show a high prevalence of H1 receptors and a low prevalence of H2 receptors. When activated by histamine, T helper 1 cells increase the synthesis and the release of interferon gamma. On the other side, T helper 2 cells have a low prevalence of H1 receptors but a high prevalence of H2 receptors. The presence of histamine suppresses T helper 2 cell function, resulting in reduced cytokine release. In humans, histamine mediates immunoregulation by binding to G-protein coupled histamine receptors. These mechanisms can vary based on the stage of cellular differentiation, microenvironmental factors, and individual genetic traits or comorbidities. By binding to specific cell receptors, histamine can induce clinical allergic symptoms. Also, it has well-documented effects on blood vessels, sensory nerves, glands, and the activation of neutrophils and eosinophils. Histamine's involvement in autoimmune and allergic diseases has been extensively studied. For example, acute urticaria and angioedema are linked to sensitivity to allergens such as foods, drugs, latex, and other substances, resulting from IgE-mediated mast cell degranulation. In contrast, chronic urticaria and angioedema arise usually from histamine release from basophils triggered by circulating IgG autoantibodies. Another example for the pathophysiology of histamine, allergic rhinitis. Allergic rhinitis is characterized by inflammatory symptoms resulting from the immune system's overreaction to airborne allergens. The exposure to a particular allergen and the subsequent release of histamine account for the symptoms associated with allergic rhinitis. For many years, H1 receptor blocker have been used to alleviate these symptoms. Recently, the use of second-generation antihistamines such as loratadine, cetirizine, and fexofenadine has increased due to their reduced sedation and greater selectivity for H1 receptors. In atopic dermatitis, 50 to 80% of children are likely to develop asthma or allergic rhinitis by the age of 5. The molecular pathways underlying pruritus in atopic dermatitis can be categorized into histamine-dependent and histamine-independent signaling. However, numerous studies indicate that pruritus in atopic dermatitis primarily depends on histamine-independent signaling. In the acute phase, T helper 2 cytokines are the main mediators, while in chronic phase is linked to T helper 1 cytokines. The interactions among T helper 1 and T helper 2 cells, dendritic cells, and keratinocytes create a complex landscape in the pathophysiology of atopic dermatitis. Additionally, histamine has emerged as a key element in the interactions between tumor tissue and infiltrating immune cells, enabling malignant cells to employ various escape mechanisms from the immune system. Also, another recent finding has highlighted the significance of histamine in hematopoiesis, as both exogenous and endogenous histamine facilitate the progression of the cell cycle in hematopoietic progenitors. Furthermore, there have been reports of agranulocytosis associated with H2 receptor antagonists such as cimetidine and H4 receptor antagonists like clozapine. As regards the clinical significance of histamine, numerous H1 receptor antihistaminergic medications are commonly used to treat various allergic conditions, including urticaria, allergic rhinitis, and hay fever. The side effects associated with first-generation antihistamines lead to the creation of a second-generation antihistamines that do not cause sedation. These newer drugs effectively block H1 receptor activation and inhibit stimulation of sensory neurons and vascular permeability, thereby alleviating symptoms of allergic conditions. The introduction of H2 receptor antihistamines has been associated with a reduction in duodenal ulcers and played a key role until the development of proton pump inhibitors. This class of medications blocks the actions of histamine at H2 receptor sites in the parietal cells of the stomach, resulting in decreased gastric acid production. H2 antihistamines have been effective therapeutic agents for various conditions, including peptic ulcers, gastroesophageal reflux disease or GERD, and dyspepsia. Common examples of H2 receptor antihistamines include cimetidine, ranitidine, and others. H3 receptor antagonists are currently being researched for their potential application in treating neurodegenerative diseases. Meanwhile, H4 receptors, the most recently discovered histamine receptors, have also been recognized as promising drug targets for therapeutic intervention in allergic and inflammatory conditions, including allergic rhinitis, chronic pruritus, and asthma. As a conclusion, the complex interactions involving histamine receptors and the cascade of mediators associated with them remain a focal point of extensive research in the medical field. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.